There is one called as a vertical gene transfer. That means when the genetic material is transferred to the next progeny. Now here we are looking at horizontal. That means when the genetic transfer occurs between the same, that is existing organisms, not in the future progeny, but in the existing organisms. So there are main 4 types, as you can see. First is transformation, transduction, lysogenic conversion, and conjugation. Now, what is transformation? It is the uptake of naked DNA. Naked DNA means the DNA that is not present within an organism but rather in the environment. So let us look at one by one. Now this transformation naturally occurs in many bacterial species like Streptococcus, Bacillus, Haemophilus, Acinetobacter, Pseudomonas. If you recall, while talking about bacterial morphology, I described something as called as biofilm, if you remember. So what is a biofilm? Slime layer mone porche, slime layer ki boleছিলাম, outer covering, that is mucoid. When many organisms come together, they form what is called as a biofilm. So biofilm, যেমন algal biofilm on water pipes or water system. So suppose this is the surface on which this is a biofilm, and there will be many bacteria, fungus, etc., many organisms within this biofilm. Now this biofilm is it's a constant structure. That means it is not a static. So organisms will come in, they will grow, they will divide, and they will take up the nutrients from that biofilm itself. And suppose some of the bacteria dies over here. So what will happen to the DNA inside the bacteria? It will come out in this biofilm, and this DNA can be taken up by this bacteria. So this is called as the naked or free DNA. It actually comes from other bacteria within the cell. Now why I have described about biofilm in this? Because this is one of the common mechanisms which is encountered especially with device associated infections. So on the devices which are placed on the human body, biofilm formation is very common, and in that biofilm, suppose there is one organism which has colonized from the hospital environment. That is not a pathogenic organism, but it bears genes which code for resistance to antibiotics. And if that organism dies, those genes or the DNA present in the biofilm will be taken up by other organisms which are potentially pathogenic. And when they cause infections, what we get is a severe or a virulent infection caused by an organism which is resistant to many drugs. Understood? So this happens in the natural environment, especially with these organisms like Streptococcus, Bacillus, and you can see Acinetobacter and Pseudomonas. These are commonly associated with device associated infections. So that is the mechanism. So these are the steps of transformation. First, as you can see, this is the bacteria which takes up the naked DNA. What happens is the nucleus splits the DNA, and this DNA enters into the cell there, and then it will integrate into the circular DNA of the bacteria. Now, a very notable experiment that was done is called as the Griffith Experiment. So what happened in that Griffith experiment is that first, in that experiment they have taken up many mice. Okay. Now this mice is an experimental mice in which Streptococcus pneumoniae, that is a capsulated strain, was injected. So what is the normal process? This Streptococcus pneumoniae will cause infection in the lung and the mice dies. Okay, infection occurs, it dies. But suppose if I have taken another mice and I have injected with a strep pneumoniae, but this one does not have the capsule. That is the non-capsulated variant of Streptococcus pneumoniae. So what happens over here? Huh? Why? Because the capsule is the pathogen, it completes the pathogenic cycle of the organism. Now, because this bacterium does not have the capsule, it can't cause the infection and the mice lives. Okay. Now, suppose in another mice, I have injected a streptococci pneumoniae that is non-capsulated, but along with that, I have added a streptococci pneumoniae which is capsulated, but that is heat killed. If I had injected only capsulated streptococci which is heat killed, it will not be able to cause infection because the organism itself is dead. But if I combine the non-capsulated variant of streptococci along with the heat killed capsulated variant of streptococci, what happens? Huh? Why? The mice dies because of the infection. What actually happened is that although we have killed the bacteria with heat, but the DNA is still there. Now, when the bacteria is killed, the DNA comes out in the environment, and this DNA has the genes for capsule. The non-capsulated variant of streptococci will take up that gene and then start forming capsule. And when it starts to form capsule, the pathogenic cycle is completed, infection occurs, and ultimately the mice dies. So this experiment proves that in nature, bacteria can take up naked DNA. Okay. So this was about transformation. Now another method by which the bacteria can acquire genetic material other than from its parent organism is called as transduction. So transduction is the method by which a bacteria acquires a genetic material from a virus. These viruses are called as bacteriophages. So suppose this is a bacteria and this is the DNA. It gets infected, say, with a bacteriophage. So how does the infection occurs? The phage will form a pore on the cell of the bacterium and it will inject its own DNA. Now the phage DNA, it can have two outcomes. It might result in two manifestations. The first manifestation is called as Porojodo. It's the lytic cycle. So what is a lytic cycle? Lytic means, lytic comes from the word lysis. Lysis means disintegration. So what actually happens is that this bacterium will, inside this bacterium, this viral DNA will start replicate. This will hijack the bacterial mechanism and will start replicate the viral genetic material. Not only the DNA but also viral proteins. It will result in formation of many small viral particles called as virions. Ultimately, when the number of virions or maturation occurs, what will happen? The bacteria will lyse and these viruses will come out. This is called as the lytic cycle. Sometimes this doesn't happen. What results is that the DNA of the virus, it does not replicate, but what it does is it ends up integrated within the bacterial DNA. And it remains as a dormant stage. When an appropriate time, depending on the environment, then it will turn into this lytic cycle. After there is a proper stimulus, the DNA will start, that viral part of the DNA within the bacterial chromosome will start replicating to form the virions and then the lytic cycle happens. So this we can call as lysogenic cycle. Again, this can also result in amalgamation of the viral DNA into the bacterial genetic chromosome directly. The bacteria is not replicated, but a small fragment of the viral DNA gets embedded within the bacterial. Why this is important from medical point of view? Any idea? In nature it is happening every time. Why is this so important? No. There are some infections which are toxin mediated. So what is a toxin? It's a protein. So this toxin, that means there must be a genetic sequence for that particular protein. This can be present on viruses and can be transferred from a pathogenic bacteria to a non-pathogenic bacteria. For example, a very common disease is called as diphtheria. Have you heard about diphtheria? Diphtheria is caused by Corynebacterium diphtheria, and this is a toxinia. That means the infection it acquires the bacteria. Exactly. So infection occurs through inhalation and it results initially in pharyngitis. The bacteria resides in the throat, commonly in the tonsillar bed. It starts producing the toxin. The local effect of the toxin is desquamation of the oral mucosa, and we can see whitish patches. That is the local effect. But as the organism multiplies and it builds up, what happens is this toxin is absorbed from the throat into the body. The toxin directly has its own effect on myocardium, that is the cardiac effects, and on the nerves, that is the neurological effect, which results in manifestation of the disease. So it is caused by the toxin, and the toxin is named as diphtheria toxin. And the funny part is, not all Corynebacterium diphtheria can cause the disease. Everyone can cause throat infection, but not everyone is able to cause the diphtheria disease. Why? Because not every Corynebacterium diphtheria has the tox gene. If the bacterium has this gene, we call it as tox plus Corynebacterium diphtheria. Okay. There are many other Corynebacteria which are similar to Corynebacterium diphtheria, which do not generally cause disease or may only cause pharyngitis. But suppose those bacteria, if they acquire this tox gene, what will happen? It will start producing the toxin and it will cause a manifestation similar to diphtheria. Have you understood? না বুঝলে বলো, বাংলায় বলছি। বোঝা গেছে? সবাই তো এমন চুপচাপ বসে যেন समझ में आया? नहीं तो हिंदी में बोलूंगा। बुझा गया? So this is the importance of transduction. Both transformation and transduction happens naturally, but from the medical point of view, transformation is very important as far as the transfer of antibiotic resistance genes from one bacterium to the other. Transduction is very important as it can transfer various genes coding for various proteins, including the toxins that are produced. Okay. The next part is very important mode of horizontal gene transfer is called as conjugation. Now, the conjugation occurs by a Pilli, specific Pilli called as the sex Pilli. So, look over here, there will be two bacteria from which one will be the donor, that is from which the genetic material will be transferred to the other, that is called the receiver. First, the sex Pilli of both the bacteria will attach and to form a channel. Now, the plasmid, first is plasmid. Conjugation, the transfer occurs with the plasmid, not with the chromosomal DNA. Now, the plasmid, what will it do? It will go to the end of where the sex Pilli has attached, and over here you can see the rolling circle mechanism happens. মনে পড়ছে কিভাবে বলেছিলাম দুভাবে rolling circle mechanism? So the after the rolling circle mechanism, the replicated DNA will cross through this bridge and enter into the new bacteria. Now both these bacteria will have the same plasmid. Now, this is very important because along with transformation, now what I have said is in transformation we need an environment from which the bacteria can take up the naked DNA which has antibiotic resistance genes. But over here, just simultaneous presence of another bacteria which has antibiotic resistance genes in the plasmids, it can transfer to another bacteria. This is a very effective method of transference of antibiotic resistance genes from one bacterium to the other in a very short span of time, and if these are placed in plasmids, it can spread very rapidly to various species and genus of bacteria. Species and genus of bacteria mean, suppose there is Acinetobacter, which is not a pathogen but has the genes in the plasmid. It can easily transfer to other bacteria also, and antibiotic resistance can spread rapidly through this plasmid. So this is the method of what we call as conjugation. Look over here, this is the generalized transduction, and the bacteriophage enters into the bacterium, then the bacterial DNA is destroyed, the viral DNA is replicated forming the virion, and then the cell lyses and it infects another cell. Now when it infects another cell, it can have three outcomes. First is it cannot penetrate, suppose it fails to penetrate into another bacterium. So we call it as abortive transduction. Or it can enter into the cell and get a stable gene transfer. That is, the viral DNA is integrated into the bacterial chromosome. So the bacteria does not die, so we call it as a stable gene transfer. And if it fails, then the lysis cycle will continue. Now what happens with the stable gene transfer is, this viral genetic material may integrate into the chromosome or the plasmid, and when the situation demands, it can form viral particles and then go into lysis. So what you can see is, during the lifespan in lysogenic part, some part of the viral DNA gets integrated into the bacterial cell. And what I have already told you, if this is a toxin producing gene, then during this lifetime, the bacteria can start producing toxin. Okay. So these are the few examples. I have already given you the example of diphtheria. Similarly, other diseases like cholera or gastroenteritis by E. coli or streptococcus pyogenetic infections, and botulism, that is caused by the botulinum toxin. So these are commonly phage-mediated toxins. Okay. Now look over here. As I have already showed you in this picture, this is the bacteria which donates the nucleic acid. It is called as the F plus, and the one which receives is called as the F minus. And what is this F? It is the donor F factor, that is the genetic material present on the plasmid. The sex pili connects both the cells, rolling circle mechanism occurs, the replicated DNA travels through the bridge, and now both the bacteria will be F plus, because both now have the F factor plus. Now there is another thing that is called as the HFR, that is the high frequency. What happens over here? This is not the plasmid DNA. This is actually the chromosomal DNA. Okay. Now when chromosomal DNA transfer occurs from one bacteria to other, we call as the HFR conjugation. Normal F plus F minus mating was with plasmid. HFR is with chromosomal. Now look at the outcome. After the transfer, the F minus, it still remains F minus. Why F minus? Because it doesn't have the plasmid. Plasmid থাকলে F plus হবে, but it does not have the plasmid, so it is F minus. The chromosomal DNA has integrated, but this bacterium, that is F minus, have not acquired the ability to transfer the chromosomal DNA to other one. That ability still remains with the donor. এটা বোঝা গেছে? আগেরটা কি ছিল? আগেরটায় প্লাজমিড যেই পেল দুটোই F প্লাস হয়ে গেল। F প্লাস মানে প্লাজমিড আছে কিনা, হোয়াট এভার মে বি দ্য কোডিং ফর দ্য প্লাজমিড। এইখানে HFR has the ability to donate the chromosomal DNA, and that DNA gets integrated into the recipient DNA, but it still remains F minus. F minus মানে F factor plasmid নেই, but it does not acquire the ability to donate the chromosomal DNA. আরেকটা অ্যাবারেশন কি F prime F minus mating. এখানে কি হচ্ছে যেটা HFR which can donate the chromosomal material, chromosomal DNA, that may be transferred to form a plasmid, সেটাকে আমরা বলছি F prime. এটা বোঝা গেছে? তাহলে ক্রোমোজোমাল ডিএনএ দুভাবে যেতে পারে, একটা হচ্ছে ডাইরেক্ট গিয়ে অন্য ব্যাকটেরিয়ার ক্রোমোজোমে ইমপ্রেগনেট করতে পারে, তার আউটকাম কি হবে F মাইনাস, অর একটা প্লাজমিড হতে পারে। Now this plasmid is not an independent plasmid, but it contains segment from the chromosomal DNA. এই জন্য এটাকে বলা হয় F prime. আউটপুট কি হবে? দুটোই F prime হয়ে যাবে। আন্ডারস্টুড? এগুলো খুব বেশি কমপ্লিকেটেড নয়, ইম্পর্টেন্স সেম। যখন তোমরা বইয়ে পড়বে অ্যান্টিবায়োটিক রেজিস্ট্যান্সও যখন পড়ানো হবে, সাম রেজিস্ট্যান্স জিনস আর প্লাজমিড কোডেড, বই লেখা থাকবে দিস আর প্লাজমিড কোডেড। সো ইফ সাম জেনেটিক সিকোয়েন্স or antibiotic resistance is plasmid coded, that means the transfer occurs through this method, and because it is plasmid coded, the transfer can occur very rapidly. But if it is chromosomal, the genetic resistance to antibiotic is present in the chromosome, the transfer can occur by this method. Now this is a slow method. Why? Because the gene has to replicate and then integrate into the recipient bacteria and form a stable replication. এটা স্টেবল হতে হবে, নাহলে তো ব্যাকটেরিয়া সার্ভাইভ করবে না। This is a slow method. But suppose if the bacteria gains the ability to form plasmid coated, তখন কি হবে? ক্রোমোজোমাল রেজিস্ট্যান্সটা ইজিলি ট্রান্সমিটেড হয়ে যাবে ভায়া প্লাজমিড। ঠিক আছে? ওকে, আই হোপ এটা তোমরা বুঝতে পারবে। ট্রান্সপোজমস মিউটেশনে পড়ানো হয়, ইনসার্শন সিকোয়েন্স ট্রান্সপোজম। ওকে, now coming to gene transfer by artificial methods. Now all these things what I've told you, all these things happen naturally. In genetic engineering what we do is use of the recombinant DNA. Even in genetic engineering we do the same thing. We identify particular gene sequence for particular protein and then try to extract it, replicate it, multiply it, and then impregnate into a different organism to get the required product. What are those? First is treatment of restriction enzymes. What are restriction enzymes? It can cut the nucleic acid. Okay. Then we can do blotting. What is the blotting? নাইট্রোসেলুলোজ পেপার দিয়ে কি করা হয়? আইডেন্টিফাই করা হয় বেসড অন দ্য লেংথ অফ দ্য জেনেটিক সিকোয়েন্স, কত কিলো বেস পেয়ার, রাইট? দেন উই ক্যান ডু আ রিকম্বিনেশন উইথ আ ভেক্টর। ভেক্টর মিনস কি বলেছিলাম আগের দিনকে? সামথিং ইউজ টু ট্রান্সফার দ্য জেনেটিক ম্যাটেরিয়াল ইনটু সামথিং, দ্যাট কুড বি আ ব্যাকটেরিয়া, কুড বি আ ফাঙ্গাস, এটসেট্রা. And finally we can do what is called as cloning. Cloning means replica of the experimental organism. It could be a bacteria, it could be a fungus. সেগুলোকে cloning. All these can be done with a recombinant DNA. Now why are these required? The first is production of vaccines. I think in the last class I have already told you how it is used. মনে পড়ছে? Hepatitis B vaccine. बांग्ला बोझो ना हिंदी बोझो? हिंदी समझ में आ रहा है क्या बोला? लास्ट क्लास में बोला था हेपेटाइटिस बी वैक्सीन कैसे बनाते हैं? वेरी गुड, चलो बढ़िया। तो So what happens is, suppose this is the hepatitis virus, for example. The antigen that is present on the virus is on the outside. Okay. Our body produces the antibody against this antigen. Now, if I introduce the virus as a whole into your body, what will happen? You will get infected. And it's a chronic and severe disease which has a disastrous outcome. So, that is not a valid way of putting the hepatitis B vaccine. In most of the cases of the viruses, what we do is we inactivate the virus. That is fine. But when the antigen is on the surface, we may not use those inactivation of the virus. What we do is we try to isolate the specific genetic sequence which codes for the surface antigen. That is the protein present on the viral on the surface of the virus. नहीं समझ में आ रहा है? वायरस के ऊपर एक प्रोटीन है, उसको सरफेस एंटीजन बोलता है. अब प्रोटीन है तो उसका कोई जेनेटिक सीक्वेंस होगा, तो हम लोग क्या करते हैं, उस जेनेटिक सीक्वेंस को आइसोलेट करते हैं. अब उस जेनेटिक सीक्वेंस को क्या करना है, आइसोलेट तो कर लिया, रेस्ट्रिक्शन एंजाइम से यूज करके हमने वो जेनेटिक सीक्वेंस निकाल लिया. अब उसको क्या करना है, उससे मुझे प्रोटीन बनाना है. अब प्रोटीन तो वैसे लैब में बनेगा नहीं, तो क्या करना है, उस सीक्वेंस को एक दूसरे वायरस में, चलो मान लो एक एडिनो वायरस में मैंने डाल दिया. अब उस वायरस के अंदर में वो जेनेटिक सीक्वेंस है. अब इस वायरस को मैंने डाल दिया, सपोज एक फंगस के अंदर डाल दिया, तो ये वायरस क्या करेगा, ये जेनेटिक सीक्वेंस फंगस के अंदर में डाल दिया और ये सीक्वेंस फंगस के क्रोमोसोम में इंटीग्रेट होगा. अब जैसा फंगस ग्रो करेगा, ग्रो कर रहा है मतलब क्या हो रहा है, वो मेटाबॉलिज्म रैपिड कर रहा है, तो मेटाबॉलिज्म के लिए उसको प्रोटीन सिंथेसिस करना है, तो उस सीक्वेंस में ये भी प्रोटीन सिंथेसिस होकर निकल के आ रहा है. अब ये जो प्रोटीन निकली, ये कौन सा प्रोटीन है, ये वही हेपेटाइटिस बी वायरस के ऊपर जो सरफेस है उसका प्रोटीन. तो हम लोग इसको बोलते हैं हेपेटाइटिस बी सरफेस एंटीजन. अब इसको अगर मैंने आइसोलेट करके किसी पेशेंट के अंदर में डाल दूंगा तो क्या होगा, इन्फेक्शन तो होगा नहीं, बट इस एंटीजन के अगेंस्ट में उसके बॉडी में एंटीबॉडी होगा. ठीक है. नाउ इट बिकम्स वेरी इजी एंड सेफ वैक्सीन व्हिच कैन बी यूज्ड. वी आर नॉट यूजिंग द वायरस एज अ होल बट ओनली अ पार्ट ऑफ द वायरस. दैट्स व्हाई हेपेटाइटिस बी इज अ सब यूनिट वैक्सीन. अब यहाँ पे जितना कुछ मैंने काम किया वो सब इसी पार्ट का है. Isolating the genetic sequence, cutting it, multiplying it, using a vector to impregnate into another organism to get the desired protein. In this case, it will be antigen. We can also use the same thing to produce antibody by what kind of antibodies? Monoclonal antibody. Only first year? Okay. So these antigens and antibodies can be used for various diagnostic tests, and these can also be used in case of therapy. Immunoglobulin can be used as a therapy. कहाँ से लाना पड़ेगा, इम्यूनोग्लोबुलिन तो प्रोडक्शन करना पड़ेगा, ऐसे ही प्रोटीन प्रोडक्शन होता है. देन ट्रांसजेनिक एनिमल्स एंड जीन थेरेपी. Okay. So what is a vector? Vector is something which can transfer parts of genetic material from one place to the other, especially in an organism. It can be a plasmid, it can be a bacteriophage, it can be a cosmid or an artificial chromosome. Now identification can be done by blotting techniques. What you have said, blotting technique is basically a gel onto a carrier molecule, that is the nitrocellulose paper, which is then on which the DNA or RNA is loaded and run against a electrochemical gradient. So it will separate out on the base of weight, kilo base pair weight. The blotting techniques are commonly 4 types: southern blotting, northern blotting, western blotting and eastern blotting. Southern blotting is used for DNA. Northern blotting is for RNA. Western and eastern are used for free DNA. Is uptake of free DNA is what is transduction? With the help of a virus. What is conjugation? Bridge through sex period. Plasmids have the following functions except plasmid is involved in all of these except conjugation lage plasmid. Okay. Can the plasmid transfer resistance? Can it transfer virulence? What is transposition? ট্রান্সপোজিশন ইজ জেনেটিক মেটেরিয়ালে একটা এন্ড থেকে আরেকটা এন্ডে সরে যাওয়া, সেটাকে ট্রান্সপোজিশন বলে। তাতে প্লাজমিডের তো কোনো রোল নেই, বিকজ দ্যাট ইজ ক্রোমোজোমাল। The Griffith experiment on mice using pneumococci strains provides direct evidence of R factor, resistance factor. Okay. So now I think I have completed the bacterial genetics. লাস্ট ক্লাসে বলেছিলাম যে স্যাম্পল কালেকশনের সোয়াবটা বাকি রয়েছে, বেশি ডেপথে যাবো না, জাস্ট ওভারঅল। Swab collection is done in case of many wounds, but at the very first let me tell you swab is a very poor quality of sample. Why? Why I am saying it is a very poor quality of sample? কোথা থেকে নেওয়া? সারফেস থেকে নেওয়া। What happens to any surface? It is a colonizing bacteria. So every time you take a swab sample, you are taking the colonizing organism, not the pathogenic organism. Hence, the chances of contamination is very, very high. That is the reason why swabs are not preferred. And swabs are absolutely contraindicated if you are looking at an anaerobic infection. এটা বলতে পারবে কেন? Because if it is anaerobic infection and you are using swab, you are already exposing the material to oxygen. So organisms will not be able to revive. Now first of all, you must understand what is a swab. Swab is a medical device that is used for collection of biological samples from human body and allows for the transport and preservation of the samples. So basically what it consists of, it's a head and a shaft. কি থাকে? This is something called as the head of the swab. This is the shaft and this is the container. Now what are the uses? First, we can use this for isolation of microorganisms from culture media. We can use for AST. এটা যখন AST পড়ানো হবে তখন দেখানো হবে কেন সোয়াব ইউজ করা হয়। Suppose I have taken a clinical sample from the swab. For microscopy, I can directly apply the swab on the glass slide, stain it and observe under the microscope. Okay. And also in infection control, swab testing is done. This will be taught to you during infection control classes. Now, swabs can be two types. One is sterile, one is non-sterile. They can be packed individually. If it is sterile, it will be packed individually. If non-sterile, then it will not be packed individually. Again, these swabs may be present along with the transport media, which may be a solid media or a liquid media. Coming to the shaft. The shaft may be made up of many material, like it could be paper, aluminum, plastic or wooden. Most commonly wooden is used and disposable swabs will have a plastic shaft. Many paper shafts are also used. Okay. Now coming to swab head. You can look at this picture. These are various types of swab head. It depends on the fiber that is used. The fiber could be cotton, it could be calcium alginate, dacron, nylon, polyester, rayon or polyurethane. Now why these are important? Because cotton swabs very commonly used, but some organisms like Neisseria or Bordetella, if exposed to cotton fibers, they die. Again, if we are looking for molecular tests, cotton fibers will inhibit the molecular, that is a PCR reaction. So if you take a cotton swab for sample, that means you cannot advise any molecular tests. Similarly, calcium alginate has got some negative points, like these are toxic for tissue cultures and viruses and for gonococci and mycoplasma. Calcium arginate swabs are not recommended for microbiological diagnosis. Next comes to the Dacron swab. It's a polyester. Now, because it's a polyester, it has got very low toxicity and molecular assays and viral assays can be easily done with the Dacron swab. তোমাদের যদি মনে থাকে কোভিড প্যান্ডেমিকের সময় যখন কোভিডে পিসিআর হচ্ছিল, কারো হয়েছে? স্যাম্পল হয়েছে? হয়নি কারো টেস্টিং হয়নি, তখন নেজোফ্যারিঞ্জিয়াল সোয়াব নেওয়া হচ্ছিল। So those were made of this Dacron and rayon swabs. Okay. Again, this swab design can be of two types. One is the traditional, another is the flocked. Traditional means what you can see in the earbuds, cotton that has been wrapped around. The advantage of this traditional swab is that it can absorb a lot of moisture or fluid. Disadvantage is that cells do not get attached to this swab easily. Mane, it can absorb the fluid, but the local tissue cells are not easily attached to it. For that, what we need to use is called as flocked swab. If you look at this picture, you can see very tiny bristles over here. So it will not be able to absorb the fluids, but when we take the sample, it takes up a lot of cells from the tissue. This is the reason why flocked swabs are more preferred in case of viral analysis. Why? Because viruses are intracellular. So you need to have the local tissue. Okay. Now commonly the swabs are used in case of pharyngitis for a throat swab or nasopharyngitis for a nasopharyngeal swab. এটা তো পড়েছি আগের দিনকে মনে হয়, how to take a throat swab. So this was all about swabs. এর বেশি কিছু জানা দরকার না। Now coming back to, I'll discuss a very little bit about Albert staining, what is being scheduled in your practical classes, and we will be given a demonstration of Albert staining. You don't have to perform it, but you will be demonstrated. Now coming back to what is Albert staining. Albert staining is a method, it's a special staining method which aims for identification of something which is called as metachromatic granules. Chromatic means colored. So different colored granules can be seen. This is a type of staining which is used to see the granules like Neisser stain or Pug stain. These can also be used as an alternative for Albert stain. Now why Albert staining is important? Because it is used to identify Corynebacterium diphtheria, especially in case of throat swab. Now why? Because Corynebacterium diphtheria produces what is called as volutin granules. What are these volutin granules? These are intracellular inclusion bodies, and complex inorganic polyphosphate get deposited in these granules in an erratic fashion. So the staining is not uniform, but rather erratic staining. This gives the appearance of a metachromatic, means various colored layer. মানে অনেক কটা লেয়ার মনে হয় ডিফারেন্ট কালারের লেয়ার আছে। This is called as the metachromatic granules. Now you must understand there are another thing called as diphtheroids. What are diphtheroids? Diphtheroids, Corynebacterium diphtheria causes diphtheria. Fine. But there are many other species of Corynebacterium also, which look similar to as Corynebacterium diphtheria, but they do not cause diphtheria. বোঝা গেছে? ঘেটে গেল। Corynebacterium নামক একটা ব্যাকটেরিয়া আছে, তার একটা স্পিসিস হচ্ছে Corynebacterium diphtheria, which causes diphtheria. Other Corynebacteria, they are commensal of the throat. So to differentiate between these diphtheroids and Corynebacterium diphtheria, we need to do this Albert stain. For example, if you encounter a patient who is having pharyngitis and there is whitish patches on the tonsillar bed, it could be diphtheria or it could be bacterial pharyngitis. For bacterial pharyngitis, you need to give antibiotic and send the patient home. But if it is a diphtheria, what you need to do? The treatment cure. You have to give antibiotics, you have to give anti-diphtheric serum because the toxin is already present in the body, and you must advise the patient for complete bed rest. You have to admit the patient and observe the patient. Patient may get into cardiac failure because of the toxin. So right then, right there, you must be able to differentiate whether it can be diphtheria or not. তার কারণ হচ্ছে ইমিউনোগ্লোবুলিন দিতে আই ক্যান হোল্ড দ্য ইমিউনোগ্লোবুলিন, আই ক্যানট ওয়েট ফর দ্য কালচার আসতে 2 দিন 3 দিন সময় লাগতে পারে। সো আই নিড সামথিং হুইচ ক্যান র্যাপিডলি ডিফারেন্সিয়েট অ্যান্ড টেল মি ওয়েদার দ্য পসিবিলিটি অফ ডিপথেরিয়া ইজ দেয়ার নট। যদি থাকে তাহলে আমাকে ইমিউনোগ্লোবুলিন দিতে হবে। আন্ডারস্টুড? বুঝতে পারছো তো? না পারলে বলবে। So for that we need to do the Albert stain. Look at this picture. This is a child. Now the child has come with a history of fever, sore throat and weakness. This can happen in other bacterial pharyngitis also. This can happen also in case of diphtheria. But try to understand the gravity of the situation. If it is diphtheria, you need to admit the child, you need to give antibiotics and you need to give anti-diphtheritic serum. And you can also see the whitish patch. So you need something which can indicate that there is a possibility of diphtheria or not. So what we do, we'll now have to take a swab from this child. So this is the process in which the tongue is pressed down with a spatula and the swab is taken from these whitish patches. Then the swab is sent to the laboratory. On a glass slide, the swab is rolled on and then stained with Albert staining. There are two types, Albert stain 1 and B. A used for staining, B for moderating, and after which we can appear the picture like this. The normal bacilli will appear with a greenish hue. The bacilli will appear greenish hue. But if the granules are present, it will appear from various color grading from green to blue to dark blue, almost brownish blue. These are the various colors that can be seen on these granules. And if these granules are seen, we can write on the report that gram-positive bacilli, because we had done gram staining previously, so a gram-positive bacilli which on Albert staining shows metachromatic granules. If this is the microscopy report, that means it indicates the child is suffering from diphtheria. Now what you have to do is, based on that microscopy report, you can't allow the child to be sent back home. You admit the child in the hospital. Depending on the body weight and the age, you give the anti-diphtheritic serum along with antibiotics and observe the patient for any complications. This is where this test comes very important. বোঝা গেছে অ্যালবার্ট স্টেনিং? So it majorly is used to identify the metachromatic granules found in disease causing organisms like Corynebacterium diphtheria. And it is also differential stain because it can differentiate between Corynebacteria which cause diphtheria and Corynebacteria which does not cause diphtheria. Okay? Last একটা কথা বলে দিচ্ছি, it is not absolute. Just because Albert stain reports are negative, gives a negative report of metachromatic granules, that does not mean the child is not suffering from diphtheria. It is only indicative. বোঝা গেছে? Many a times it may happen that the case was actually indeed of diphtheria, Albert stain negative may come. Medical science কোনো কিছু absolute নেই. So this is only indicative. Okay?